All right, let's start. So, in the last class, I did a lot of stuff. So, I did all of this, right? This is chapter number 2, by the way, of the IMAT syllabus. I did this part: cell theory, cell size, prokaryote, eukaryote, animal and plant cells. I also did viruses, all of this. I did that. I did transport across the membrane in the last class. Also did cell structures and their specific roles. This is basically the different types of organelles. I also did the functions of them. And today we are going to finish this chapter and we'll start with cell cycle and cell division, and then we'll move on to chromosomes along the chapter. So, uh, in this chapter, you learn a lot of the cell division part, which is mitosis and meiosis. So, let's start with chromosomes first. Let's start with some definitions first. So, uh, DNA packaging, right, is when DNA wraps and coils around histone protein. Why does this happen? To reduce space. Remember that DNA would combine with histone protein so that it reduces space. And when these two combine together, they form something called nucleosomes. They form something called nucleosomes. And many of these nucleosomes, they combine together to form chromatin. So DNA plus histone makes nucleosomes. A lot of these nucleosomes combine together to make chromatin. Right. So during cell division, DNA undergoes further coiling, which results in tightly coiled DNA known as chromosomes. Now, this chromatin will undergo further coiling and it will become chromatin, chromosomes. So, we start with DNA, which combines with histone. It makes nucleosomes. Nucleosomes combine together to make chromatin, and a lot of these chromatin they coil together to make chromosomes. Right. So, what is the structure of chromosomes? Chromosome has several thousand genes. And what is a gene? A gene is a DNA which codes for protein. Gene is a part of DNA which codes for a protein. And locus, right, is a position of genes in a chromosome. So this gene has some position on a chromosome, let's say here or here. And the position of this gene is called locus. So locus is position of gene on a chromosome. And if organism is from the same species, the genes are of the same type. And alleles may be different in same species. So what is an allele? Allele is a different form of one gene. Allele is a different form of one gene. Remember this, locus and genes. Remember their definitions. Now, what is a homologous chromosome? A homologous chromosome is pairs of chromosomes found in diploid cell. I'll explain what diploid and haploid are. For now, just know homologous chromosomes are found in diploid cell only. And one chromosome is from the father and one is from the mother. And diploid cells will have two chromosomes or 2n. And uh, they have similar centrosome position. What is a centrosome? I'll explain that as well when I get to the chromosome definitions. So similar centrosome positions. Chromosome, right, have similar centrosome positions. And what is a centrosome? A centrosome, why is this? Whose video is on? Can you switch off your video? All right, so moving on, centrosome, right, is the center position at the center where one chromatin and one chromatin comes together, right? So when these two chromatins come together, the center is called the centrosome. And remember, homologous chromosomes will have similar centrosome positions. So the positions of the centrosomes would be the same in homologous chromosomes. And homologous chromosomes have the same genes but different alleles. Again, allele is a different form of one gene. So what is a sister chromatid? A sister chromatid during interphase, right, DNA replication happens in the nucleus, and one chromatid becomes two identical chromatids. So what is DNA replication? DNA replication is when one chromatin, right? This is a chromatin, would become two sister chromatids. And the thing that is holding these two chromatids together is the centromere. The centromere holds two chromatids together, and the number of chromosomes equals the number of centromere. So the number of chromosomes will be the number of centromeres that you have. So let's get to the main part. That was just the basics. Now we are going to the main part. So what is mitosis and what is meiosis? So mitosis, right, gives you diploid cells. What is diploid cells? Diploid cells are when you have 2n as the number of chromosomes, or basically two chromosomes. Whereas haploid cells, right, have half the set of chromosomes, or the number of chromosomes are n, or 1. Now, mitosis, you get full set of chromosomes from the parents, and you make somatic cells. Mitosis is for somatic cells. What are somatic cells? Somatic cells are basically the cells in our body. So like liver cells, muscle cells, kidney cells, all of that. So mitosis makes somatic cells, and they have full set of chromosomes, and they form diploid cells. And at the end of mitosis, right, you will form two uh, two cells. Whereas in meiosis, you will form 4 cells. And these 4 cells will have half the set of chromosomes. So let's say that I started with this cell and it underwent meiosis. And if this cell had 2 chromosomes, then at the end of meiosis, each of the cell produced will have 1 chromosome. So the number of chromosomes gets halved. So meiosis happen in sex cells. What is sex cells? The cells that are called sperm and egg cells. So sperm and egg cells are sex cells, and meiosis is the division which is involved in sex cells. Right. So first I'll do mitosis and then I'll do meiosis. So why is mitosis important? Because mitosis maintains the number of chromosomes, and new cells can retain its function. So I told you earlier that mitosis gains the full set of chromosomes, meaning if a cell which underwent mitosis had 2 chromosomes, at the end of mitosis, the cells produced would also have 2 chromosomes. So mitosis produces full set of chromosomes, and this is important in maintaining the cell's function. So let's say I had a liver cell, right? And this liver cell it gets old and it will undergo mitosis. When it undergoes mitosis, the liver cell will produce the same set of genes and chromosomes that it had. So the liver cell will maintain its function. So at the end of mitosis, I form 2 liver cells, and these 2 liver cells will have the same function as the original cell. And mitosis is also important for the growth of multicellular organisms, not unicellular. I'll get to why unicellular is not important. The important is multicellular. So mitosis is important for growth of multicellular organisms. Basically, for you to grow into a good human, into an adult, you will have to go a lot of mitosis, not meiosis. So human growth is because of mitosis. Another important part of mitosis is tissue repair or cell replacement. When you get a cut on your skin, mitosis will be undergone, and you will replace the cut on your skin. So you will get new skin cells because of mitosis. The skin around the cut, right, would undergo mitosis, and you will get new skin cells. So cell replacement or tissue repair. And finally, asexual reproduction. This is not really that important, but this is another function. This is a separate topic, by the way. So asexual reproduction is also very important. So 4 reasons why mitosis is important. Learn that. And now I want to explain the anatomy of chromosomes. So chromosome, right? This is a chromosome, by the way. And this part right here, this is chromatid, this is one chromatid, and this is two chromatids. So on a chromosome, I have two chromatids. There are two chromatids on a chromosome, and the ends of this chromatid, right? These ends on the chromatids are called the telomere. They are the end of chromosome region. So the end of these chromatids are called telomeres, and I'll get to telomeres after I finish cell division. But other than that, the center is called the centromere. Uh, and the centromere job is to hold uh, hold the chromatids in place. So they hold the chromatids in place. Right. So let's get to mitosis now. So mitosis, right? Mitosis has four stages. Mitosis has four stages. And in total, the cell division of somatic cell has four stages as well. So let's get to the stages, right? So this part right here is one stage. This is another stage. This is another stage, and this is another stage. So the M phase, right? The M phase is called the mitosis, and it's only a part of cell division. Mitosis is only a part of cell division. There's three other stages of cell division as well. It's the G1 phase, the S phase, and G2 phase. And first comes the G1 phase. What is G1 phase? G1 is where you grow cells and synthesize proteins and other substances for cell division. So obviously you need energy for cell division, and in this part you will synthesize proteins and the stuff that you need for cell division, basically ATP. So G1 phase is where you start to grow the cell and synthesize proteins and other substances. This is the first part of cell division. And then comes the S1 phase. When you finish G1, you go to S1 phase. S S phase or S1 phase is where you undergo DNA replication. And when you do that, it will result in sister chromatids. And this is basically S phase. DNA replication will result in sister chromatids or new chromosomes, right? So in a cell, right? So cell is not undergoing cell division. This is all of this, right? G1 and S phase is happening inside the cell. It's not undergoing cell division right now. It's preparing for cell division. So S phase is where you replicate DNA and you will get sister chromatids or chromosomes. And finally comes the G2 phase. And after the G2 phase, you will undergo mitosis or cell division. So G2 phase is where you will also undergo growth of the cell and you'll duplicate the centrioles. I explained what centrioles is in chapter 1, but not in detail. I'll explain what it is. But for now, in G2 phase, you will undergo duplication of centrioles and you will form two pairs of centrosomes. Two pairs of centrosomes. What is a centrosome? A centrosome is when two centrioles combine together at right angles or 90 degrees. So this is a centriole and this is another centriole. They combine together to form centrosomes. And in G2 phase, you'll also undergo repair of DNA replication error. So if there is any error in DNA replication, G2 phase is where you will fix it. And this phase is also where you prepare the cell for mitosis, right? This phase is where you prepare the cell for mitosis so that it will go to mitosis. So this was the three stages. And then comes the main stage or mitosis. So I told you earlier, there are four stages of cell division: G1, S, G2, and then M phase. And remember that this part, right? This big chunk where there is G1, S, and G2 phase, this three stages, the G2, the G1, and the S phase, right? This is called interphase. These three stages are called interphase. And then comes mitosis. So after interphase comes mitosis. So mitosis is like a separate stage that has a lot of phases. So mitosis or M phase itself has 4 different stages. And then you will go to cytokinesis. So let's go on to mitosis, right? So again, I told you earlier, mitosis has 4 stages. So what are the 4 stages? Prophase, metaphase, anaphase, and telophase. Right. So mitosis has these 4 stages. Before mitosis comes interphase. After interphase, you'll get these 4 stages. So first step is prophase. Prophase is when the chromatin or the chromatid would uncoil. So uncoiling of chromatid would condense and become shorter and thicker. So in a cell, you'll have a lot of chromatin, and all of this chromatin will start to uncoil and become thick and condense. It will also get shorter. So what you'll see under a microscope when you're undergoing prophase is these large chromatin that are uncoiled. So spindle fiber forms which starts attaching to the centromeres. What is spindle fiber? Spindle fiber is formed by centrioles. Centrioles or centrosomes. Centrosomes are two centrioles again. And in cell division, you will get centrosomes. You'll use centrosomes and not centromeres. So each end, right? Each end has centrosomes. And each end has centrosomes or two centrioles, and they will form spindle fiber, which will start attaching to the center of the chromosomes or the centromere. The center of chromosomes are called centromere. Centrioles or centrosomes attach to the centromere, and centrosomes move to opposite poles. So these centrosomes, right, the centers will start to move at the opposite poles. Now, the nuclear envelope breaks down. The nuclear envelope inside the nucleus, around the nucleus, sorry, would break down, and nucleolus, which is inside the nucleus, will also break down. Now, this was prophase. So in a summary, in prophase, you will uncoil the chromatin. Chromatin becomes short and thick, and spindle fiber starts to form by centromere, and centrosomes will move to opposite poles. So nuclear envelope breaks down, nucleolus also breaks down. So before I continue, I want to explain two things. Centrosomes and centromere. This is very confusing. I also get confused a lot. I think in my lecture, I got confused here as well. So I want to clear this confusion. Centrosomes are two centrioles at right angle. Whereas centromere is the center of chromosomes. And centromeres hold the chromosomes together. And the centrosomes also produce spindle fiber. And this spindle fiber, right, will attach to the centromere. So the spindle fiber produced by the centrosomes gets attached to the centromere. Remember the difference? I saw a question where they were asking the difference in these two things, and a lot of the times you'll get this wrong. So memorize this part. Now, first stage of mitosis is done. I'm done with prophase. Now comes metaphase. What is metaphase? The centrosomes, right? The centrioles will reach the opposite ends. So in prophase, the centrioles were moving to the ends of the cell, but now they are done. They are at the ends. They've reached the ends of the cell. So spindle fiber are fully formed. Now the spindle fiber are fully formed, and the chromosomes line up at the metaphase plate or the center. So this is the metaphase plate. You can this like this or like this, and they will either form here or here at the metaphase plate or the center. So chromosomes line up at the center, and chromosomes attach to the spindle fiber at the centromere. Chromosomes attach to the spindle fiber at the centromere. Again, spindle fiber will attach to the centromere of the chromosome. This is basically metaphase. So now that the spindle fiber is attached to the centromere, right, we'll go to anaphase. We've done with metaphase. Spindle fiber is attached to the centromere or the chromosome, and then comes anaphase. So anaphase, right, is when the centromere of each chromosome divides. So I have this cell, I have the chromosomes line up at the equator, I have the centrioles or the centrosomes, right? Centrosomes are basically two centrioles, and they've attached to the to the uh, to the chromosomes or the centromere. So what the centrioles do is they split the chromatids, and they will break the center or the centromere. So the centromere of each chromosome divides, and the spindle microtubules or the spindle fibers would now shorten. So this spindle fiber, right, you see, will become shorter. They will become shorter and shorter by every stage. So, for example, this is one chromatid, and this is another chromatid. Before it was like this, but now they've broken apart and they move to opposite ends. Chromatid moves to opposite ends. And at this stage, you need ATP to pull the spindle fiber. So in order for the centromere to pull the chromatin, you have to need ATP. So you need ATP to shorten the spindle fibers. And the chromatins will move to the opposite ends. So this was anaphase. And in telophase, right, in anaphase, the chromatids are still moving. They've not reached the end, but they're moving. And at telophase, the chromatids will reach the poles. So they will reach the ends of these poles. So at telophase, this is telophase, the chromatids, right, which were split, would reach the end of these poles. And at this stage, the chromatid would start to uncoil. What do I mean by uncoil? The chromatid will become big, large, and thin. So they will become large, long, and thin. And in this part, the nucleolus will reappear, the nuclear envelope will also reappear, and the spindle fiber would break down. So basically, telophase, right, is the opposite of prophase, because in prophase the nucleolus was disappearing. Here it's reappearing. In prophase the nuclear envelope was disappearing. Here it's reappearing. And also the chromatid was condensing, but now here in telophase the chromatid is uncoiling. So telophase, you've reached telophase, and what happens after telophase is cytokinesis. Now, for me to explain what cytokinesis is, I'll go to this diagram right here. So this is mitotic cycle basically. I want to zoom in. So first was prophase. Prophase is when the chromatids become condensed, they become short and thick. The centrioles or the centrosomes, they will start to move to the ends. In metaphase, the centrosomes are fully at the ends, and the spindle fiber are fully formed, and the spindle fibers attach to the chromatids or the chromosomes. And then in anaphase, the centrosomes start to pull the chromosomes so that they move to the opposite ends. And in telophase, they move to the opposite ends, and you will start to form the nucleolus and the nuclear envelope. So the nucleolus, nuclear envelope will start to reappear here. Here in prophase, it was disappearing. In telophase, it's reappearing. So after telophase comes something called cytokinesis. Now, cytokinesis is when the cell completely divides into two new cells. So I had one cell which was undergoing all of these stages: prophase, metaphase, anaphase was happening in one cell. But cytokinesis is when this one cell will become two. So the cell divides into two. So cytokinesis, right, is when the animal cell undergoes division and the cytoplasm divides into two. So the cytoplasm divides into two daughter cells. Now, this the part where you start to form this division, right? This part where you start to separate the chromosomes. The cytoplasm is called cleavage furrow. So cleavage furrow is the part where you will form this line where they'll separate. So cytoplasm would start to separate. Now, this was my cell division in animal cells. In plant cells, there's not a lot written about plant cells, but what you need to know is that in plant cells, everything happens the same except the cytokinesis part. So in plant cell, vesicles are transported at the center instead of this normal cytokinesis. Instead of cytokinesis, this part will happen. This part right here would happen after, instead of cytokinesis. So vesicles transported at the center of the cell to form a cell plate. Cell plate is basically this line with vesicles. So the cell plate becomes cell wall. This vesicle becomes cell wall. And then the cytoplasm divides into two and organelles are shared out. This is very basic and you only need to know this much about plant cell. So the vesicles at the center, they form cell wall. This cell wall before is called cell plate. They form cell wall and cytoplasm divides into two and the organelles are shared out. And this is mitosis. So I explained mitosis and now I want to go to telomeres. So what is telomeres? Telomeres are the end of chromosomes, and telomeres are very important because their role is that they prevent loss of genes at the tip of each chromatid. So every time the chromosomes divide, to prevent the loss of genes, the telomeres will prevent this from happening. You don't know how it happens. You only need to know the role, which is they will prevent the loss of genes. And telomeres has a problem, which is that telomeres get shorter with each mitotic division. So every time the cell undergoes cell division, so like this liver cell undergoes 5 cell divisions, right? So every time the telomere will get shorter. And this causes them to stop doing their job. What was their job? They would prevent the loss of genes. Because they are getting shorter, they stop doing their job, and this will cause the cell to age and stop functioning properly. So cells stop dividing at one point where the telomeres are very short. So when the telomeres get too short, right, the cell will no longer undergo cell division. And that's the point where you start to age. So as you age, right, your cells will divide, but there will be a age like 40 or 45 where your telomeres of each cell is too short to divide. So you can't undergo cell division anymore and the cells will stay where it is. So this will cause aging, the cells will stop doing their function and the cell will eventually die out. Very sad, but this is life. So to prevent this, there is an enzyme called telomerase, and telomerase is an enzyme that prevents telomeres from getting shorter. So telomerase is an enzyme which prevents telomeres from getting shorter. But this unfortunately is not present in animal cells. In healthy functional animal cells, you don't have telomerase. So the telomeres would keep getting shorter. That's the reason you age. Now, why am I explaining telomerase? Because telomerase is present in cancer cells. So cancer cells has telomerase, and that's the reason cancer cells can divide unconditionally and they can divide forever. So because they have telomerase, they can divide a lot, and they basically divide forever because the telomeres would not get shorter. So stem cells, right? Let's move on to stem cells. Stem cells are undifferentiated. What do I mean? Undifferentiated basically means they are unspecialized. So they have no roles. They don't have any specific role other than the fact that they would simply divide into two cells. So stem cells are able to continuously divide via mitosis without stopping because they have telomerase. So other than cancer cells, telomerase is also found in stem cells. So telomeres do not get shorter, and each cell cycle will keep happening without the risk of telomeres getting shorter. So stem cells, they divide and they produce two cells. One is another stem cell and one is a specialized cell. So what is the role of this specialized cell? The specialized cell that is produced by stem cell can have multiple roles. So this specialized cell can be used for cell replacement, tissue repair, growth, or basically uh, you can make red blood cell. Basically, any cell can be made by stem cells. So stem cell can make a specialized cell for specific roles like liver cell or blood cell, red blood cell, white blood cell. All of this can be made using stem cell. Now, very short note is this thing called stem cell therapy. So stem cells are introduced into new adult human, and this promotes healing of injury and disease. For example, leukemia. Leukemia is, you'll get to leukemia at the last chapter, psychology and anatomy, but leukemia is the cancer of white blood cells. So to stop this cancer, you can use stem cells and make new white blood cells that are functional. So in short, stem cells make specialized cell that can be any cell of the body, any somatic cell, and they also make another stem cell when they divide by mitosis. Right. So that was basically mitosis. And in the next class, I'll start with meiosis because I only have 4 minutes left. So I want you guys to solve this question. I had a question in mind. So this question right here, I think it's from 2016. You have 2 minutes and then I'll do it on my own. So this is related to mitosis. This question is about mitosis. Yeah, you have 2 minutes. Solve it. Someone said E. I'll wait for more answers. Right. So I don't know if the answer is E. I'll check. Let me check if the answer is E. Someone is saying D. You know what? Let's do it on our own. Let's see what's the answer. And then we'll check the mark scheme. So the table shows some events of the normal eukaryotic cell cycle or cell division. This in eukaryotic cells, you undergo mitosis to make new somatic cells. And so which row shows the correct sequence of events from left to right as they occur during part of the cell cycle? So basically, What they're asking is how mitosis happens. So someone said E, right? Let's see E. E is row 3. So step 1, chromosomes line up at the equator. This is metaphase. This is called metaphase. And then spindle fiber shortens. Yes, this also happens in metaphase. And then nuclear envelope reappears. This happens in uh, telophase. This happens in telophase. And then the cytoplasm divides. This is cytokinesis. So this seems like right because it's following the right sequence. So metaphase, then telophase, then cytokinesis. Yes, it's not in sequence as in that you're skipping anaphase, but the sequence is right. So anaphase, my bad, guys. This part is anaphase. I forgot that the part where spindle fiber shortens is called anaphase. So metaphase, anaphase, telophase, and then cytokinesis. Because anaphase is where spindle fiber shortens, metaphase is where the chromosomes are lined up at the equator, nuclear envelope reappears in the telophase, and cytoplasm divides in cytokinesis. Right. Yeah, anaphase. I'm sorry, it's anaphase. So this seems like right to me, but someone said so. For now, E seems right, but someone said D. Let's do D. Row 4, right? DNA starts to condense. Yes, this is prophase. Nuclear envelope disappears. This is also prophase. Spindle fiber shortens. This is anaphase. Chromosomes line up at the equator. This is metaphase. This is wrong because anaphase comes after metaphase, not before. So this is wrong. So the right answer is D. Sorry, E. Row 3 is the right answer. Yeah, I can do the rest, but I know the right answer, so why worry? So, yeah, this is the class. In the next class, we'll do my meiosis. I hope you learned something. Bye. See you in the next class.